Oh, so thank you for inviting me today here. I'm excited to join this meeting for the first time. It's good to see other people who are excited about ketamine, suspicious. It was great to have fun yesterday at the debate and see different views. So on the debate, I agree it's not cancer. It's actually probably worse than cancer because people kill themselves because they're depressed. So going back to our clinical trial, I will start with the financial disclosure. None of these is relevant except this one. So mainly Yale and the VA have a patent application related to one of the clinical trial that I'm going to present today. And as you may expect, this clinical trial take a lot of effort. So these are some of my colleagues that helped with these studies. These studies were done both at Yale. I'm currently at Baylor. These were funded by VA and DoD and Pfeiffer Foundation. So the first clinical trial is in depression, ketamine in depression. And this was mostly not focusing on efficacy, or this was a mechanistic trial trying to look at the role of mTOR in depression. And this based on elegant preclinical work. Probably you've heard about some of it during this conference, mainly the hypothesis that depression is related to loss of synapses in the brain, especially in the prefrontal cortex. So here in this cartoon, we depict like synapses under normal condition and then synapses after depression and chronic stress. In the animal, you can see that you have loss of synapses, lower density of these synapses. And what remain there is relatively, they are less strong synapses. It turns out that ketamine and other antidepressants reverse this synaptic disconnectivity. And the cool things about ketamine that they actually reverse that robustly within 24 hours of treatment in the animal. And based on the animal models, the main hypothesis at the time is that this increase in mTOR signaling here is a critical for increased synaptic plasticity after administering ketamine in healthy animals. And we thought that's pretty interesting. And we aim to demonstrate that in humans. So we did a clinical trial, mainly mimicking with the preclinical data. In preclinical data, you give ketamine, you show that ketamine increased plasticity, or you give rapamycin before ketamine, and you show that rapamycin blocked this increase in synaptic plasticity 24-hour post-treatment, and block what looks like antidepressant-like effect in rodents. So we wanted to do that in human, and we designed the study as close as possible to the animal data. And here, relatively, it's a simple crossover design. And so one thing about ketamine that probably most of you know, that the effect don't last long. So within 2 weeks, most of the patient usually relapse, and then you can cross them over to the second treatment. So here we have a patient, he drink orange juice, and then 2 hours later, get ketamine, or drink orange juice that has inside of it rapamycin, therapeutic dose of rapamycin. And then they will get the ketamine, and the hypothesis, the 24-hour post-treatment, the pre-treatment with rapamycin will reduce the antidepressant effect. And this is randomized, so some people will start with placebo ketamine, some people will start with rapamycin ketamine. And this is mainly what we found. So I'm going to walk you through this figure. So the red is showing the MADRS score, which is the severity, following the placebo ketamine session. And the blue is following rapamycin ketamine session. So what we see here, people relatively have moderate to severe depression. Hours after treatment with ketamine, especially at 24-hour, you have significant reduction of ketamine, of depression severity. And then 3-5 days later, up to 2 weeks, you have a slow relapses. And by 2 weeks, this is not significant compared to pre-treatment, but they still have numerically a relatively lower severity. And this is what we observed after pre-treating them with ketamine, with rapamycin. So at 24-hour, there is no difference. And if anything, is actually rapamycin is numerically lower. So we failed to show the preclinical data support in human data, mainly that rapamycin did not block the antidepressant effect. But the surprise is actually what we noticed later. We see that people who got rapamycin, actually, they weren't starting to relapse as fast as we were observing with placebo ketamine. So here the slope, the start of the relapse is after 24 hours. Here the start of the relapse is after 5 days. One thing to keep in mind based on the half-life of rapamycin is we think that rapamycin was having strong anti-inflammatory effect up to 5 days. So we were seeing this with the total severity, and it's kind of more impressive when we look at response rate and remission. So here at 24 hours, we see no difference in response rate between red, which is placebo, and blue, which is rapamycin sessions. Same with the remission. So mainly we cannot support the preclinical data with these human data. But the surprise is at 2 weeks, if people were on placebo, most of the patient relapsed, only 13% did not relapse. But this is triple the effect if you took rapamycin before that. So here we have 3 times that response rate at 24 hours and 4 times the remission rate, at 2 weeks. So we start kind of scratching our head. Why do we have this kind of unexpected results of tripling response rate and quadrupling remission rate 2 weeks after treatment? So we first look at the psychotomimetic effect of ketamine. So if we look at CADS, these are measures of dissociation and positive and negative symptoms following ketamine administration during the infusion. So we'll see kind of similar to what we'll expect. This is both sessions together. So mainly ketamine will increase dissociative symptoms, and these will go back to normal within 2 hours after treatment. If we look at them separately, we see that after placebo, you have increase in dissociative symptoms, and the same after rapamycin. There were no significant difference in anything. That's actually the dissociative effect were lower after rapamycin. Keeping in mind, there were some data suggesting maybe the extent of this dissociative effect will predict the antidepressant response, mainly more dissociative effect during infusion, predict better response. So this was not supported by this data. Mainly rapamycin did not worsen the dissociative effect, did not worsen the positive symptoms, and did not worsen the negative symptoms. Another thing to rule out, maybe rapamycin have different effect on anxiety. And here we show that the anxiety effect, anxiolytic effect of ketamine were the same following placebo and the same following rapamycin. Another thing to rule out, maybe there were drug interaction between rapamycin and ketamine. Maybe somehow rapamycin increased the dose or the level of ketamine, and that's why we have a prolonged effect. First, we didn't predict this because they work differently in the liver, and actually we can show that the ketamine level following placebo is the same following rapamycin. If anything, it was even lower following rapamycin, not higher. One more thing to look at is, did we reach therapeutic level with rapamycin, giving it 2 hours before treatment? So the therapeutic level for the immunosuppressant effect is 5 to 10. And here we see that prior to giving ketamine, we actually were at 26. And at 4 hours after ketamine, we're still at 10. So we definitely reach peripherally the therapeutic level that we were targeting. And the last thing, there are hypotheses in the field that the more you have inflammation, the better you will respond in terms of antidepressant effect to anti-inflammatory drug. And we looked at ESR and CRP, kind of crude measures of inflammation, and we showed that they have no effect on the antidepressant effect that we found at 2 weeks. So mainly we started with one question, one hypothesis, spent 3 years, a lot of work, and we ended up, as usual, with multiple questions and even more hypotheses. So the first question is, why the discrepancy? Why we have different results from preclinical to clinical data? The second question is probably the most important. What's the potential mechanism? In general, why people relapse after we give them ketamine? Why they don't stay good forever? Why we don't cure them after we give them ketamine? And the last question is, what's the impact of this unexpected finding? So for the first question, I think one thing to keep in mind that in the preclinical data, they actually put the rapamycin in the medial prefrontal cortex, which we cannot do in humans. So we give it peripherally. So theoretically, in the animals, you block mTOR in the prefrontal cortex, and you keep it this normal in the rest of the brain, which we couldn't do that in a human. And the other potential explanation, these animal models are really healthy animals. They were stressed out. They're not like depression where it's episodic, you have a genetic component, and you have many different pathology that what you observe with the chronic stress. So the potential mechanism, one thing is really, as a field, we didn't spend a lot of time on is why people relapse after we give them ketamine, why they don't stay better for a longer time. And so one hypothesis that we're proposing is that the depression has a priori pathology. And I kind of, to simplify it, I'm going to present it in a simple way. Imagine that the hypothesis that depression is related to synaptic plasticity is actually correct. And then patient before a treatment, they have high severity, mainly lower synapses, mainly the higher neuroinflammation leading to elimination of these synapses. And at pre-treatment, they have low synapses in the prefrontal cortex and more severity. You treat them with ketamine. Ketamine significantly increase synaptic density in the prefrontal cortex, and this reduce it at 24 hours. But in patients, you still have the same pathology that you started with, that neuroinflammation is going to start eliminate the synapses that were created by ketamine. And then at 24 hours, you start this relapse where you see there's a slope of increasing of severity after treatment. When we treat them with a strong anti-inflammatory drug, perhaps we delayed this elimination by 3 to 4 days. And then we're observing the relapse is starting after day 5 instead after day 24. The second hypothesis is that the rapamycin itself may have antidepressant effect. And actually, there are preclinical data showing that it does have antidepressant effect. But these data were following treatment for like 2 weeks, more chronic treatment, not just acute single treatment. And that hypothesis that it's working through autophagy, which is regulated by mTOR signaling. And the third question is, what's the impact of this finding? Are we like kind of witnessing a third strike of serendipity? We know that first with TCA and second with ketamine, both were relatively unexpected finding. And are we witnessing kind of third and unexpected finding that will show us different way of treating antidepressant? And definitely the time will tell that. And hopefully more studies will be done to confirm or to negate these findings. So that's the first for ketamine in depression. The second clinical trial is ketamine in PTSD. And this is trial mainly focusing on efficacy of ketamine in PTSD. And as you know, ketamine worked for bipolar depression, for unipolar depression, and everybody was saying, why you're even wasting your time doing this study? It's going to work for PTSD. And we thought, good, this is easy study. We'll do it. We'll show it works for PTSD. And then this was mainly done at the VA to encourage the VA to support treating veterans with PTSD with ketamine. And it didn't work, but I'm going to tell you more about the results. So we did this study. This study was randomization 2 sites in veterans and in active duty. We looked at the regular standard dose 0.5 versus placebo versus low dose 0.2 milligram. And we treated patients twice a week for 4 weeks. And to encourage people to stay in this study, we offered open label for those who did not do well during the double blind. And so this is the main finding. Also on the left, we'll see the PCL score, which is the self-report of PTSD severity in these patients. And here we see the treatment period. And unfortunately, the primary outcome were negative. Mainly, we didn't have interaction between the study arms in terms of response for PTSD. And I'm going to kind of walk you through each dose separately. So the standard dose is the 0.5 that's often used for depression. And then we will see here at 24-hour post-treatment, people actually get better compared to placebo. So the black is the response following placebo, and the red is the response following ketamine. So at 24-hour, you have a significant improvement compared to placebo, but this improvement was short-lived. So these at day 3, 7, and so forth, these are pre-next infusion. So this is all 3 days following the previous treatment, and 3 days following the previous treatment was mostly comparable to placebo. The interesting part that was unexpected is with the 0.2. So this study, we start working on it in 2012. And we really didn't have much data about 2 doses versus 3 doses, or if ketamine works in PTSD or not, or what's the 0.2. But over the past 10 years, we have so far 2 clinical trials showing that 0.2 milligram does not work for depression. And here we see, again, it doesn't work for PTSD at 24 hours, but over time, they continue to get better. So actually 0.2 in a post hoc analysis, it did do much better than placebo over time. And this is another measure, which is clinician administered, which is we did at the end of treatment to confirm the self-report. And mainly the results are similar to the self-report. There is no interaction, but if you look separately, the 0.2 was significantly different compared to placebo. The 0.5 was better, but at a trend level. And then we asked the question, how long these effects, if any effect, were there? How long did they last? Because we followed people for 4 weeks after the last treatment. And then we see with the 0.5 was only a trend at the end of the treatment, and there were no significant over the follow-up. Versus with the 0.2, you have significant difference at the end of the treatment. And this is significant retained for 4 weeks post-treatment. So mainly, the effect of the 0.2 after 4 weeks of treatment did last much longer. So the question is, what about the antidepressant effect in this population? Most of these patients have a major depressive episode and has high severity of, like you see here, it's kind of similar to what we're seeing with the treatment-resistant depression. And by the way, most of these patients also treatment-resistant depression. We require them to be treatment-resistant to at least 1 drug, but many of them have way more than 1 drug treatment-resistant. So the antidepressant effect actually was significant at the 0.5. Mainly, you can see still antidepressant effect in this population at the 0.5, and the 0.2 was not significant, kind of confirming a previous clinical trial that 0.2 is not working for depression. So what's interpretation and kind of future direction? Why do we have this results? One thing to keep in mind that this is a military population, and historically with previous study, military population tend to have more treatment resistant. They actually, PTSD study that looked with SSRIs, they have a lower effect size, and many of them failed to show the effect of SSRIs on PTSD symptoms. The other things is the pathology. Perhaps the pathology with PTSD is different than depression in terms of response to ketamine. One thing to keep in mind that even with bipolar depression, the effect of ketamine were very short, that people relapse within 3 days, and there is no significant difference compared to pre-treatment. And perhaps with PTSD, we're seeing something similar, that the effect is very short-lived. The second one to look at is that the low dose seems to be working on PTSD, but this is, keep in mind that this is a secondary analysis. It's really need to be replicated prospectively to be more confident of this finding. And there were another small study by our colleague at Mount Sinai where they give 3 times per week for PTSD, and it did work. So potentially, future studies should look at more frequent ketamine administration if they want to administer kind of ketamine separately. And also maybe we can try to prolong the ketamine effect or make it more by giving rapamycin or other anti-inflammatory drug. And the last thing to keep in mind that really the finding with PTSD are not as consistent with the finding with depression. So our study failed to show the primary outcomes, but there were other studies also that failed to show the effect of ketamine on PTSD symptom. For example, Adriana at Mount Sinai in the first study showed significant effect at 24-hour post-ketamine in reducing PTSD, but this was not the case in her second study. So it looks like definitely the ketamine effect on PTSD are less robust and more inconsistent. And thank you.
So we're open for questions. Yes, please.
Did you happen to collect any subjective responses, and apart from the the MADRS findings or the
yeah, so we have others. Usually we have the self-report, like the quiz, and they're consistent. For the PTSD study, the main outcome was a self-report, which is the PCL, mainly because the CAPS is usually administered once a month, and we were interested in the 24-hour effect. So the primary outcome for that study was the self-report with the PCL. We confirmed it with the CAPS. But usually, most of the other measures that we took are in the same direction. For example, for the PTSD, we have the GAT-7. All of them were in the same direction. They did better with the 0.2 than with the 0.5. The antidepressant, they did better with the 0.5, not with the 0.2.
Sorry, I also meant with qualitative subjective feedback. Like we heard earlier this morning about some qualitative findings. Did you collect any?
We did collect with the PTSD, and this will be tested separately. We did these were just mainly we did the primary outcome so far, but we have other measures. We have cognitive, we have effect on life. For the PTSD also, we allowed people to have alcohol problem, but they have to be just sober. So we're going to look also kind of secondary measures if that was affected as well.
Hey, Chadi, great job. Quick question regarding, one of the big concerns has always been in the PTSD patients that the reports of, increased anxiety and things like that following getting ketamine could be a negative effect. Did you notice, was there any difference in, you've treated a lot of them?
Yeah, so at the time, Jerry, as you know, at this time we were thinking about the study. One of the concern is it safe to actually give ketamine in PTSD, and that time, and that's why we selected the 0.2 as a lower dose. So because we know the side effects are dose dependent, so the lower dose will have less side effect. If 0.5 wasn't tolerated, maybe the 0.2 will be better toleration. So we didn't have any issues with the 0.5. It was actually similar to what we're seeing with antidepressant. In depression study is very well tolerated, and we didn't see any relationship between the extent of anxiety during infusion or psychotomimetic effect or dissociative effect during infusion and the final outcome. Because one of the thought is maybe 0.2 is working better because it's better tolerated, because they did have much less side effect with the 0.2. It's barely noticeable, but that was not the case.
So thanks for that presentation. The rapamycin study is phenomenal experimental medicine study, really great. Do you think that just shows that ketamine doesn't act through the mTOR system?
Well, we couldn't support that. We definitely cannot support that, but we cannot rule it out. So we have to keep in mind it could be working in the preclinical data. What was done is really inhibiting mTOR in the medial prefrontal cortex and keep it intact throughout the brain. So it could be a network effect. When you give it peripherally, the expectation that you're going to inhibit mTOR equally throughout the brain. And so what we interpret this result, first, that we could not support our preclinical data. We could not support that. Second, that perhaps it's more, you need a network effect. You need to affect mTOR differently throughout the brain. So that's mainly the extent of how we can support it. What we think about why we have this result, that perhaps we kind of unexpectedly came across a different way of treating depression, which is you treat depression with antidepressant, like in this case, ketamine, and you prevent relapse with anti-inflammatory drug, which is the hypothesis that maybe this, if we were ever to get funded to do follow-up on this, that will be the second thing to mechanistically show that by having anti-inflammatory effect for a longer time, we can prevent relapse for a longer time. And that would support the hypothesis that relapse is related to anti-inflammation.
So the ketamine can act generally, but the mTOR rapamycin has to be specifically localized.
It looks like it's a network effect, has to be localized, because other preclinical data where they give it also peripherally, it did not block the synaptic plasticity as well. So it has to be a network effect in way of modulating mTOR. And then one thing to keep in mind, that's network effect, like I know like there are drug being developed to target mTOR and have antidepressant. And this might still work, because even with ketamine, we give NMDA antagonist, but there is a network effect where throughout the brain you have different extent, like in the prefrontal cortex is actually the opposite. You're stimulating the glutamatergic system, not blocking it. So with the development of mTOR, it still could work if the affinity of the drug or the way it's administered eventually is going to give a network effect on the brain.
If you were going to put a grant application to test that hypothesis about inflammation, what drug would you use? Would you use rapamycin, or is it the sort of drug that actually is too toxic to use repeatedly?
Yeah, so that's a good. So for follow-up, one thing is we're interested, and then if we can convince the NIH to fund anything ketamine. But so mainly the interesting part, so we're seeing a very nice parallel between the time rapamycin is working and the relapse. So one possibility is to stick with everything is the same, so we can replicate what we found before, and have people take just 1 dose 2 hours before the first infusion, or take 2 doses 2 hours before the first infusion and on day 3, because then we can push it another 5 days. And then this way, it's still 2 doses. We're not worried about the toxicity. We're not worried about anything. And we're not changing too many things to be able to interpret. And the good thing about, even though like rapamycin itself is a dirty drug, like ketamine, so it may be having other things other than mTOR. But if we can prove that this giving it repeatedly, it's a neuroinflammation related, then there are tons of drugs. Actually, one of the best successes with drugs is with the modulation of the immune system, where you have all these with biologics and all these successes. So if it boils down that prolonging the anti-inflammatory effect will prolong there are multiple drugs that could be tested. There are other drug mTOR inhibitors that has less side effect and more tolerated than rapamycin. There are regular anti-inflammatory. If we know exactly which with the immune system is relating it, you can develop for that. So they're mainly the options are endless if we can show this mechanism is working.
PTSD population cohort seems to be quite diverse, so it's sometimes difficult to look into signals. Did you look into antidepressive effects of ketamine to your PTSD cohort? Yeah,
so the antidepressant effect has significant antidepressant effect was 0.5. So the antidepressant effect in this severe military population were maintained. And it's similar to other clinical trial in depression. It worked with 0.5, it did not work with 0.2. But the finding with the PTSD is a PTSD specific. PTSD severity didn't get better with 0.5 for a longer time. It did get better after 24 hours of the first infusion.
Thank you for a good presentation. The rapamycin study, how many patients were there? I missed that, sorry.
So this is about 20 patients. It's a crossover. So
Type 2 error?
It's possible. One thing to keep in mind that most of the studies with ketamine were actually less than that. We were like the first study was almost 8 subjects to 10 subjects. Their application was 16 subjects. And these studies benefit from a crossover design. So you in the same person, you're crossing them over to the second. So it's possible. That's why you need replication. because it's just one study. So it's a relatively on the larger end of crossover the studies, which is 20 subject and 20 subject within subject 2 weeks later will repeat it. But definitely you need the replication before making any kind of strong conclusion. Nobody should start treating patient in the clinic because of this data. We really need to do more study to demonstrate efficacy. And for safety, I know this is the main question is usually how safe it is. Both ketamine and rapamycin have been in the field, and we know they're safe with those one infusion. But we want to see if, like, down the road, if we want to give it more often. So it looks like based on this data that rapamycin would be required like once a week or every other week. So you want to see how that in terms of safety, how safe it's going to be in this population. Rapamycin is actually usually taken chronically, daily dose, and it has a lot of side effects and usually used for cancer and renal transplant. So it does have side effects. And if that turns out the case, that's required multiple times, once a week, we want to see the extent of those side effects. Are they going to be similar to once a day or much less?
So can I?
Yeah.
So your assumption is that ketamine is like fast acting and transient. But this is also based on very small studies.
Well, there is a lot of studies right now. So this is the PTSD study is 160 subject. The collective, the antidepressant effect, yeah, it's a short-lived. Usually most of our patients will, if you don't give them anything else, you'll just give them, you'll enroll treatment-resistant, depressed patient, you'll just give them 0.5 milligram, a single dose. You can expect that 80% of them will meet the relapse criteria by 2 weeks. And this is consistent across the studies, in multiple studies, not only in our data. People do relapse after a single infusion if they're treatment-resistant depression. That's why it's being given like twice a week and multiple times.
How was the blinding in the rapamycin study? Were people able to tell the difference between the two?
Yeah, so for the rapamycin was open label, so everybody knows they're taking ketamine because they get ketamine. The blinding is for the rapamycin. So to our knowledge, there is no way to know because it's just they're going to drink a cup of juice on both days, and one of them has rapamycin. And in our like asking people, does this taste differently? It's just orange juice. So it's a very good blinding for this test design because most of the difficulty with the blinding is with the ketamine because people will figure it out. We actually have good blinding also with the PTSD because most of the people thought maybe they're on the 0.2. So in the ketamine PTSD, we asked people if they can guess what arm they're on. Placebo, 0.2 and 0.5, and they couldn't, nobody guessed it. Most of the people said the 0.2 pretty much. And so it was statistically different.
Are there any questions online?
Well, I have a couple from myself.
Yes, please, absolutely.
People are hungry, we're holding them back.
4 minutes to lunch.
One, the ketamine and the rapamycin. Do you think that is the combination of the two that this prolonged effect, or are you envisioning your next studies like giving them ketamine and then trying to maintain this antidepressant effect, just giving them?
Yeah, so our primary hypothesis that I'm betting on is that is actually rapamycin. It did not induce antidepressant effect. It prevents relapse, and this is mainly based on observation. And then once we do the study, either we'll support that. And this is actually the good news. If we can support that, then you have a lot of options how you can use different drug than rapamycin, because rapamycin is a pretty heavy drug. And then you have a lot of options for anti-inflammatory drug. And it's in the field, like the neuroinflammation and inflammatory has been very exciting. Unfortunately, it didn't pan out as antidepressant so far. So this could be a totally new way of treating depression. It's not giving anti-inflammatory to have antidepressant effect, but to maintain the antidepressant effect of mainly kind of a relapse prevention.
Yeah, the question of like, if you study, did you see any effect on the symptoms after? PTSD?
Yeah, we didn't see, we were hoping to see maybe like there's a subclusters where they did better. And that was not the case. Mainly the finding at the cluster were similar to the finding in the general. The 0.2 did much better in the long term at the end of the treatment. And actually they stayed better 4 weeks later with the 0.2. Okay, lunchtime.
Yeah.
Thank you.
I'd like to thank you again.